Welcome to Phi Moments, a podcast by Quantiphi.
Hello, everyone. Welcome to Phi Moments, a podcast by Quantify, where we explore AI breakthroughs shaping enterprise transformations. I'm Rahul Ganar, and today we will be diving into innovations redefining drug discovery and development. Joining us are four exceptional leaders from the world of technology, pharma, and scientific research, each bringing in a unique lens of how we can move forward faster, safer, and more human centric in healthcare and life sciences. The way drugs and vaccines are being developed is changing faster than ever. Stem cells based platforms, when combined with advanced AI, are opening entirely new possibilities. A DART platform built together with Quantify and Transcell Biologics is a strong example of how we can deepen our understanding of biological systems and disease pathways without relying on the traditional methods. When you layer AI on the top of these stem cells technologies, you start unlocking insights that helps predict safety and efficacies much earlier, significantly accelerating the drug discovery operations. Beyond stem cell systems, we are also seeing a larger momentum toward non-animal next generation methodologies that are bringing in to reshape preclinical testings. With regulators increasingly support these approaches, and overall the industry is at this point of time adopting more of these technologies. It's not just promising, it's actually a necessity. For today's episode, our focus is on building next-gen platform for preclinical testing workflows. And to explore this theme, we are joined by three incredible leaders who bring in perspectives from technology, pharma, and scientific innovations. Please join me in welcoming Asif Hasan, co-founder of Quantify, an award-winning AI digital-first company. With over 2 decades of experience in applied AI, data science, and cloud computing, Asif has helped leading healthcare and life sciences organizations translating their cutting-edge AI into practical solutions for drug development, preclinical testings, and clinical developments. His work emphasizes building scalable platforms, improving predictive accuracy, and aligning innovations with real-world and regulatory needs. We also have Dr. Sadanand Vodala, a life sciences technology leader with nearly 2 decades of experience advancing preclinical and clinical developments, and as well translational research and digital transformation across several pharmaceutical organizations. He has led cross-functional programs spanning biomarker strategies, clinical development, and data platform organizations, helping teams integrate scientific insights into technology to accelerate overall drug discovery, development of especially oncology assets, and improve decision making. We also have Dr. Subhadra Dravida. She is the founder and CEO of Transcell Biologics, a stem cell researcher turned into entrepreneur. Subha earned her PhD in industrial biotechnology and has built Transcell to commercialize stem cells based models, biobanking, and non animal preclinical tools. She is deeply invested into translational science, bringing lab discoveries with tools that can reduce dependency on animal testing and accelerate oral drug development. And to guide this discussion, we are fortunate to have a very senior guest moderator today with us. Joining us as a moderator, Debraj Dasgupta, a senior executive with extensive experience across strategy, commercial, and technology functions in life sciences. He is also currently on the board of advisor with Transcell, and we are fortunate to have him with us today. Debraj brings unique perspective to collaborate science, business, and innovation, making him the perfect person to lead this conversation. Debraj, thank you very much for joining with us today. The floor is yours to take us into the discussion. Thank you.
Thank you, Rahul. Across the industry, we are witnessing a turning point. Regulatory bodies are beginning to signal openness to non animal models, and pharma organizations are under pressure to deliver safer, more effective therapies faster. At the same time, AI and digital platforms are maturing to the point where they can finally add real value in complex scientific workflows, not just as pilots but as integral tools. From my vantage point, one of the biggest shifts is the move from proof of concept science to enterprise adoption. Companies are not just asking, can this work? They're asking, how can we scale it responsibly, integrate it into decision making, and align it with regulatory pathways? That's the real challenge and also the real opportunity. Today, we'll explore these opportunities and barriers, and we'll hear how next gen platforms like DART and other non animal testing models are shaping a more predictive, ethical, and efficient preclinical pipeline. So, let's begin by grounding ourselves in the state of the industry. So, Asif, if I can start with you, what are the biggest barriers you see in applying AI to biological systems like non animal NAMs? Is it data quality, heterogeneity, lack of scale, or something else?
Sure. First off, thank you all for organizing this amazing session on a very, very important topic. And it's my pleasure to be on this highly esteemed panel. Coming to your question, Debraj, I think it's a very critical question because on paper, this kind of marriage looks perfect, right? AI is great at pattern recognition, predicting patterns. Biological systems provide the complexity, but in reality, I think we're hitting a lot of friction as I see. If you look closely at the landscape, it's not just one barrier. I feel it's a triad, three specific challenges from my vantage point. First is the consistency chasm. Second is what I call the small data trap. And third is the regulatory black box. So let me unpack it one at a time. So, first up is what I call the consistency chasm. This used to be about data quality, but it's much deeper than that now. It's about heterogeneity. So, in the world of non animal methods, whether you're looking at organoids, human microphysiological systems, or high content imaging, the data is actually just incredibly fragmented. So you might have one lab in Boston doing transcriptomics, another one in Berlin doing imaging, and these are two completely different data types. But even within the same data type, we have a problem with what the whole idea of artisanal biology, because the protocols aren't standardized yet. Every lab grows their tissue slightly differently. When you feed that varying data into an AI model, the model struggles to generalize. And it doesn't just learn the biology, it also learns the noise of that specific lab protocol. So until we bridge this chasm with standardized metadata and consistent ontologies, our AI models are going to be highly variable. The second barrier is what I call the small data trap. And this one is about scale. We hear about big data all the time, but in the NAM space, data is actually quite expensive and slow to generate. So we aren't training foundation models of the entire internet with billions of parameters. We are often training on data sets of 50 to 100 experimental runs. Maybe we are boosting it with synthetic data, but it's still very small and it leads to a lot of overfitting. Plus, we have a missing data problem because labs rarely publish failed experiments, the negative data. But AI needs both sides of what, not just what looks safe, but also what looks toxic. And without that negative data, models are flying actually half blind. So finally, even if we solve the consistency and scale, we hit the hardest wall, which is the regulatory black box. So this is an issue of trust and explainability. So regulators like FDA or EMA have a mandate to ensure patient safety. They cannot simply accept a machine learning algorithm that says, trust me, molecule is safe. They require mechanistic transparency and they need to know why the model made that prediction. And right now, deep learning is notoriously difficult to interpret. So we are still building the validation frameworks to prove these digital prediction map accuracy to human biology, not just to the animal models we are trying to leave behind. So it's data quality, yes. Is it scale? Yes. But mostly it's about lack of unified language between biology and code. So that's sort of the triad of barriers that I see in applying AI to biological systems like non-animal methods.
Thanks, Asif. So, Sadanand, if I can come to you next. So, as someone coming from the industry, from a pharma lens, what are the critical features you want to assess when evaluating non animal NAMs from a drug development stage perspective?
So thank you, Debraj, for that question. And I echo Asif's sentiment here. Thanks for having us on this panel. This is an exciting topic of discussion. And this is going to change the way the industry does drug development. Right? There's a long way for us to go. And you raise a very important question and a point here. FDA is generating new guidelines or providing new guidelines on a regular basis. Even just a couple of days ago, there was a new release from FDA on NAM and non-animal model testing. What kind of evolution is going to happen in these guidelines from FDA is something that I'm going to closely watch, because at the end of the day, we as a sponsor, from a company point of view, we are responsible for the safety of our investigational medicine product, either preclinically and or clinically. So we have to justify to FDA, here is a drug, this is safe, we want to test this in a clinical setting and or launch it from a commercial point of view as well. So the onus lies on the sponsor, right? So having said that, the things that I would be closely watching in the next few years would be what kind of guidelines that FDA is going to provide to us. How do we align with FDA on these guidelines? What kind of NAMs can we align on in terms of generating the required data for safety assessments and or clinical assessments? And there are some existing models that will come into play, like real world evidence data. Is that going to be sufficient? Is that going to be relevant from an IMP, investigational medicine product point of view? Those are still questionable here. Data from organoid models will be useful. Computational tox approaches, while speculative, will need validation as well. So there's a lot of things happening in this field. I'll be closely watching a lot of these things. Couple things that also come to my mind is, how do we align with FDA on these NAM models? What is the validity of these models, the reliability of these methods? Are these NAM models generating the sufficient data that we need? Are they equal to the existing animal testing that we currently do? Are they better in terms of their data generation? Those are some things that would be very high on my list of priorities, as well as we transition from animal tox or non-primate tox studies to NAMs in this case. Of course, the old epithet that, time is money, it comes into picture here. The cost for us from an industry point of view, the amount of money that we spend to do preclinical work is huge. So the cost is a big picture that comes into mind. Scalability of these NAM models, I think, is also very important. Typically, what we do on a preclinical side is we take a lead molecule, go test it on non-primate studies, right? But can we do several molecules and investigate these several molecules simultaneously using NAM approaches? I think that's something that I would look forward to as well. That way, we can generate more safe molecules, that as opposed to taking one lead molecule and study the safety of our molecule. And the time component is also very important here. We know the non-primate studies take a long time to generate the preclinical data. Hopefully, the new NAM models that we, with FDA and the CROs that would be involved in this, the time will be compressed to shorter time to get to clinic. That's something that I would be closely watching as well. Those are a few things that come to my mind. I'm happy to elaborate based on how the discussion progresses.
Thank you. Subha, if I can come to you next. So as a scientist founder who developed DART, what gaps in traditional preclinical models did you see that convinced you that this type of innovation was urgently needed?
Sure. Thanks, Debraj. And I am as well super excited to be in this panel. As a practicing scientist, I have identified, three major shortcomings in traditional preclinical models that present significant challenges, according to me. The first one being the exorbitant costs, lengthy timelines, and reliance on ethically contentious animal based systems that are still prevalent. And then the second one is the use of transformed or genetically modified saline based in vitro models, which have been around for a long time, but definitely, fail to provide reliable insights. Then the third one is a contract testing system that lacks consistency, where pharmaceutical assets are sent to external laboratories for all detailed evaluations. Given my background as a dedicated translational biologist in drug discovery domain, particularly with expertise in induced pluripotent stem cell technology, it was only logical to focus on developing non-animal alternative drug testing strategies, I must say. During then the COVID-19 pandemic, this commitment evolved into a drive to create DART, a deployable enterprise solution that combines clinical surrogate biological models with AI, uh, offering tangible benefits to the end users. Devraj.
Thank you. So that gives us a strong foundation and very different perspectives to this discussion. Now let's talk about what it takes to move from potential to practice. How do we build trust? What does adoption look like at scale, for example? So Asif, if I can come back to you, how do you build AI models that remain robust and explainable enough for use in safety or toxicology decisions?
Yeah, sure. I guess, Debraj, what you're asking me is how do you build an AI that is smart enough to predict toxicity but transparent enough to trust with human lives? And of course, that's a multi-billion dollar question, and even bigger ethical implications are there. And that's the reason why Dr. Subha and myself and others at Quantiphi and Transcell are working behind it and very excited about it. It's really a safety paradox. Deep learning in the natural state is a black box. It's a great prediction tool, but terrible explanations. But in toxicology, of course, trust me, doesn't work. So to solve this, you can't just throw more data at the problem. You have to change how you apply the model. So at Quantify, we believe the solution has a three-layer architecture. We call it number one, the hybrid engine, number two, the why, and number three, the guardrails. First, you need a hybrid model. Old school AI just looked at chemical structures, like taking a picture of a key and guessing if that'll open a door. The new approach is biology informed. So you don't just feed the AI the chemical structure, you feed it the biological context. You integrate historical toxicology records with omics data and known biological pathways, and this grounds the AI. And it's not just finding the mathematical patterns, but also kind of learning the underlying biological plausibility. It understands that structure A triggers pathway B, which leads to liver toxicity. So it makes the model robust because it's reasoning like a biologist, not just kind of like a mathematical calculator. The second is the why layer. Here you need to understand the mechanistic interpretability. This is where we open up the black box. We use post hoc explainability tools like SHAP values or attention mechanisms, but you tune them for the toxicology. And instead of the AI just saying, this drug is toxic, it highlights the specific parts of the chemical structure responsible and the links to a known adverse effect profile. So it says, I predict toxicity because this chemical group interacts with this specific protein pathway. And this traceability is non negotiable, of course, for the regulatory bodies. And finally, you build that regulatory guardrail. So this is about defining what we call the applicability domain. You don't let the AI guess on things it hasn't seen before. You rigorously validate it against benchmark data sets and set up that continuous monitoring. So if the data drifts, the model recalibrates. This is exactly what we are doing with the DART framework. So we've combined these three layers: robust human relevant data, explainable hybrid models, and rigorous validation to create a system that offers full traceability from the data to the end prediction.
So we've talked about features, adoption, and what kinds of evidence are most persuasive. If we build on that, if I come back to you, Sadanand, beyond preclinical trials, how do you see your data from non-animal models translating into the way clinical trials are actually run? For example, trial design, patient safety monitoring, or decision making once a drug reaches testing in people.
Debraj, this is an interesting question, partly because AI is lurking around the corner and wants to make a significant impact in the way clinical trials are run now, right? And then how do we combine AI and the NAMs into clinical development of various assets? I think, having said that, we're thinking about step 8 when we are at step 3 or 4, right? With these two big points in my mind, I'll try to touch base on few examples of how NAM models can be used even in the context of clinical development of assets. First thing that probably comes to my mind is how do we generate human dose estimates? There's a huge disconnect between our preclinical work that we do and the first in human dosage that we have to define before we get into the clinic. When an IND is submitted, we have to tell FDA how are we going to do human dose estimates, what would be the starting dose. We do a lot of computational modeling, like quantitative systems pharmacology modeling. I think some of these NAM models would be very, very helpful in doing human dose estimates and of course the human dosing assessments. And then continuing along the same theme, the dosing regimens can also be tested in the NAM models very easily because dosing regimens, step-up dosing, monitoring safety, increasing, decreasing dosage based on what is developing in terms of a signal, both on a safety and also in terms of pharmacodynamics, it would be very, very beneficial in that space as well. There's also a huge disconnect sometimes between what we observe in the preclinical setting and when we get into clinic, it opens up a new black box for us in terms of safety. The unforeseen, unexpected toxicities that we observe in the clinic can be quickly investigated using NAM models, I think, because to go back to animal testing and spend 6 months to a year to do that animal testing, it's very time consuming. By that time, as a sponsor, we may or may not be thinking about continuing the molecule through clinical development. If we can quickly use some of these NAM models to understand what are these unforeseen toxicities in the clinic, why are we seeing these tox, can we investigate the reasons for it and develop mitigation strategies along those lines? I think NAMs would play a significant role in that space as well. And along the same lines, again, there's always patient-to-patient variability in terms of safety and efficacy that we observe. What are the underlying reasons for that? How is patient X different from patient Y? That is another space where NAMs will play a significant role, or we can develop NAMs in that space as well. And I think one final point that comes to my mind would be, historically, we have done animal testing on healthy animals, for example, right? But in the context of any disease, the disease itself alters the entire systems pharmacology or systems biology in a subject, right? You're testing these drugs in healthy subjects or healthy animals, whereas the drugs are being given to patients whose biology is completely altered, right? So developing generating some of this data from these animal models and or disease relevant NAMs would be very, very crucial, not only to understand safety, but also to dosage regimen, dose estimates. A lot of these unforeseen challenges that we see in the clinic can be addressed using NAMs. I think these are a few things that come to my mind. It depends on how we develop NAMs in this space to address these paradoxical questions that we see in the clinic.
Anything to add from your side, Subha?
Yes, in line with what Sadanand has just shared, to highlight the relevance of our DART solution, especially during the clinical development phase, particularly in oncology, we excel at recreating the ex vivo tumor microenvironment through physiologically relevant in vitro or ex vivo models let me just elaborate. One of the demonstrated key applications of DART is its support for ex vivo clinical trials, especially for novel immunotherapies, where patients are recruited, but the test drug isn't administered directly. Instead, it is applied to the petri dish with patient-sourced tumor samples utilized and recreated tumor microenvironment in the labs. In fact, this process allows us to conduct even routine PK and PD assessments, and along with any safety-related evaluations, ultimately, leading to the reporting of clinically relevant insights. Then another example in the same context that I can quote is the Comprehensive In Vitro Pro Arrhythmia Initiative. This FDA endorsed in vitro method is specifically designed to test cardiotoxicity during phase 1 clinical trials. Likewise, DILI, drug-induced liver injury assessments, are another classic example. It's actually a clinical parameter, also built on human-induced pluripotent stem cell in vitro technology to support clinical development of drugs. So there are few very established and classical cases, like we can take as examples where non-animal NAMs can play a key role in clinical development stages of the drugs.
Thank you. See, we'll all agree that trust and adoption are critical, but they only matter if the science itself is ready, right? So, Dr. Subha, if we dive into the innovation at the core of the discussion, now let's talk about DART. So, in your experience, how do the advanced human cell models in DART compare to conventional lab systems in terms of scalability and relevance?
Sure. So, Debraj, first on the relevance part, DART focuses on human microphysiological systems, which are comprehensive configurations that integrate sometimes organoids or even sometimes 3D models with additional components to replicate the specific physiological environment of the system in question. When I say a specific system, I mean those related to the human hematopoietic system or immune system or nervous system or even cardiac systems. Both the existing literature and our findings show that HMPS configurations can closely mimic the natural microenvironment and at the same time can offer high scalability, especially when sourced sustainably. So that's a point. Our experience highlights that specialized biobanks which expertly handle human biological discards, are vital enablers of scalability with in vitro systems. Now, moving to the second aspect of scalability that DART offers, it lies in the implementation of bioassays. Here, the use of customized AI and machine learning neural networks acts as the foundational biological model, in fact, setting us apart in this field that we believe. So, just to kind of summarize, this innovative approach addresses all the identified challenges, positioning DART as the cornerstone of non-animal animal methods, with a flair of human biology that democratizes the very strategy itself of testing safety and efficacy.
Thanks, Subha. In order to make it very specific, can you share an example where DART has already delivered insights or outcomes that traditional approaches could not?
Sure. In fact, this is the best question to me. See, since DART has two components to its design, the first one being an in vitro or ex vivo platform with relevant human biological milieu, with the second one as the AI-enabled in silico platform for data analysis. Some of the drug assessments to do with, let's say, again, DILI or repeat dose toxicities, and also sometimes detecting anti-drug antibodies or PD biomarker signature identifications and using it as basis for demonstrating bioequivalence like functional readouts, including finding new functions of old molecules, are unique module-based outcomes that traditional approaches cannot just beat the DART relevance that we bring to table. And also as DART employs both multimodal and multimodal architectures for comprehensively analyzing wet lab data. Some of the other bioassays, such as the best examples being, vaccine neurovirulence and immunogenicity testings, are the critical readouts delivered from the end users' ongoing workflows as they adopt it as an enterprise solution. But for the DART strategy, according to us, obtaining human mechanistic insights in the most cost and time efficient manner is otherwise not possible through any traditional approaches, Debraj.
Thank you, Subha. So these are very good examples, and now we have a good grounding on what's possible today. But now the next question is, where do we go from here, right? What does the future of preclinical testing look like in the next 5 years? So if I can come back to you, Sadanand, do you believe that digital and human relevant platforms are ready to be used as central decision making tools in drug development, or are they still more of a supporting tool alongside traditional methods?
Debraj, this is another exciting question as well, especially considering, as I alluded to earlier, AI is lurking around, and FDA is willing to work with pharma sponsors to move away from animal models. And how do we integrate a lot of these different aspects into drug development? Having said that, and Dr. Subhadra gave some fantastic examples of how these NAMs can be utilized in the context of clinical development, both on the preclinical and clinical development as well. I see there is a lag between the new technologies or the NAMs that are coming into the foray of drug discovery and development to adoption of these technologies, right? We as, sponsors, individuals, a little reticent to change. And especially in the field of science and discovery and development of compounds and drugs, we want to see validity and proof of concept before these models can be applied and utilized in our day-to-day drug development strategies. So I think there is that lag. There needs to be a lot of education to bridge this gap between the advent of these new technologies and how pharma companies can utilize them for our drug development strategies. So we are not there yet, right? We are not there yet, especially from a drug development point of view. How much of NAM models have we used in the past 2 years, 3 years for our big decision making in terms of should these molecules move into clinic? Should these molecules have these molecules generated enough safety data versus using the traditional models and the NAM models? We haven't shown that bioequivalency, as we say, between these two different approaches, the old models versus the new. And we're also sitting in the phase of precision medicine. A similar philosophy kind of probes me in that direction as well. Each challenge that I see, each drug that I develop is going to give me new questions. And how across the board are these NAMs valid for my different therapeutic modalities and or T-cell engagers, bispecifics, monoclonal antibodies, ADCs. Can these NAM models be applicable to different therapeutic modalities? Those are questions that pop up as well. So having said all of these things, I think it's an exciting time. We are not there yet, but the future is very bright. Pharma companies are willing to invest in this space because FDA is willing to work with the pharma companies, and it reduces time and cost and money and resources for us, right? So we are in an exciting time, but not there yet.
Anything to add there, Dr. Subhadra?
Yes, I know Sadanand's point of view is he says that we are not there yet, but then the future definitely looks good, of course. And then in continuation to our experience with the DART implementation science, I just would like to highlight a specific use case here involving testing of anti-sera potency through in vitro neutralization on the human hemato, neuro, microphysiological system. In fact, this is a peer-reviewed approach. And then this approach has not only aligned well with data from animal models, but also provided a potency range that indicates clinical application doses. So this is, according to us, one of the best case studies where digital clubbed with the human relevant platform is proposed as a replacement to the traditional and notorious animal based method in this particular domain. But then yes, I mean, like this, there are more examples that are evolving as we speak. And of course, data speaks. Like, I mean, evidence is what everybody is looking at, and we will be there. And yes, it is going to be wonderful for adopting non-animal NAMs.
Thank you, Sadanand. Thank you, Dr. Subhadra. Very clear. So, Asif, if I can come back to you, looking ahead, how do you see AI evolving over the next 5 years to accelerate adoption of human relevant platforms like DART across the industry?
Yeah, let me start off by acknowledging the viewpoint from you, Sadanand, in terms of like not being there fully, at this point in time. I also share the sense of optimism that Dr. Subha has because of my personal proximity to it. And see, I think for adoption of these technologies, sometimes we need a forcing function. Like, for example, the pandemic was a forcing function for mRNA vaccines, right? So, Devraj, when someone asks me to predict the state of AI 5 years from now, I have to pause because honestly, I think the pace of innovation that we've seen is so staggering that even looking 2 years out feels like trying to gaze into an entirely different era. And that's kind of contributing to my sense of optimism. I have a clearer view of the next 18 to 24 months, so I'll restrict my answer to that time horizon. I see two fundamental shifts that are going to completely rewrite the playbook on how we do science from a drug discovery, drug development perspective. The first is going to be the emergence of kind of, the concept of digital twins, but in complex biological systems. We will likely see that we'll be moving into a world where biology was a purely a wet lab problem to one where it's becoming more of a computational problem. And I'm not just talking about simulating a single cell, I'm talking about modeling complex inter-organ interactions. So imagine using an AI to analyze organoids derived from human stem cells, replicating the actual tissue architecture of a liver, a heart, or a kidney. We are getting to a point where we can detect toxicity mechanisms that animal models often miss because, quite frankly, mice aren't, humans. So this allows us to fail fast, fail cheap in the digital world, so we can succeed safely in the physical one. And the second shift, and this is the one that truly excites me more, is the move towards inverse design driven by autonomous research agent. So in the old world, we used kind of sequential testing, screening millions of existing molecules, hoping to find one that worked. It was efficient, but still it was reactive. In the new world, we are kind of flipping that script. So we will have AI agents fed on diverse physiologically relevant data sets from human tissue biobanks. These agents won't just screen, they will design molecules from scratch with specific safety profiles already baked in. So you tell the agent, I need a molecule that targets this protein but avoids this toxicity pathway, and it kind of engineers one uh, in a reverse way for you. We are seeing some of this type of work in our generative chemistry research labs. So in that sense, I feel that DART represents this kind of new era of animal-free toxicity testing. It's a hybrid approach that combines the speed of AI screening with the biological relevance of the advanced human data sets. And by shifting this work from cages to code And leveraging platforms like Dart, I feel we aren't just saving millions of animal lives, or we are also accelerating time to market, and we are kind of acting with a deeper sense of compassion. And we are reducing our reliance on animal testing, improving patient safety simultaneously. So that to me is kind of like the ultimate promise of this technology, and I'm hopeful that in the next 18 to 24 months we will see some very credible demonstrations of this, in not so distant future.
Thank you. Thanks, Asif. Thank you for all your perspectives. Great discussion. It's very clear. And as closing, I would like to say that while challenges remain, the path forward for human relevant AI enabled platforms is both exciting and full of potential. With that, I'll hand back to Rahul to close us out.
Thank you, Debraj, and a big thank you to our panelists Asif, Dr. Sadanand, and Dr. Shiba for bringing in such thoughtful and practical perspective on the table today. What we've heard really uncovers that future of preclinical testing is not just about accelerating the timelines or reducing the cost. It's about building far more human-relevant models using AI responsibly and effectively, and aligning with regulatory environment that is increasingly open to non-animal approaches. At Quantiphi together with partners like Transcell Biologicals, we committed to pushing this vision forward through the platform like DART, keeping scientific rigor strong, improving patient outcomes, and ensuring innovation continues to move in the right direction. And finally, thank you everyone who joined us today. We're excited to keep this momentum going on. Join us next time as we explore the next wave of innovation shaping life sciences landscape. Thank you very much.
Thanks for tuning in. To learn more about how we're helping enterprises transform with AI-first solutions, subscribe to Phi Moments on your go-to podcast channels or visit quantiphi.com.